RNA. What's the usage of the non-RNA? So there are several types of the application about non-coding RNA. Here we showed like 4 common types of non-coding RNA. So first type, it's the non-coding RNA can bind with tRNA. And then second type, this non-coding RNA can bind with mRNA. And the third type of non-coding RNA, this type of RNA can bind with the protein. And then the last type would be non-coding RNA will bind to a small molecule. So there's even some kind of a non-coding RNA. It can bind to the different target. Like for example, you can bind to 3 different type of a protein. So we would say this type of non-coding RNA is called a multiple binding site RNA. And then since this non-coding RNA, they can bind to the different target. They have like diverse set of functions. So the first type of the function is called scaffold. What's a scaffold mean? So which means this type of the non-coding RNA contain binding site for a multiple component, such as a group of different protein. Much like the bins in the building, a non-coding RNA can act as scaffold for formation of the complex, like for example here. So this non-coding RNA can bind into 3 different type of protein, and then this whole thing, it's a complex, right? So the usage of the non-coding RNA in this case would be help to forming the complex. And then the second application of non-coding RNA, it can guide a molecule to a specific location in a cell. For example, a non-coding RNA may bind to a protein and guide it to a target site in the DNA that is part of a particular gene. So like, for example, this non-coding RNA will bind to this protein, and then after they form the complex between the non-coding RNA and the protein, and then this non-coding RNA can guide the protein to a site in the DNA by the binding affinity between the non-coding RNA and the DNA, right? So basically, it's guide this protein to reach to the place of the DNA. And then another type of application of the RNA, it can change the protein function or its stability. So after this non-coding RNA binding with the protein, it can change the property of the protein. And then before, like for example, original those protein has no function of being catalyst. But after formation of the complex of the non-coding RNA with the protein, so this complex can work as a catalyst. And then another application would be ability of protein to bind to other molecule. Right here is a good example, because non-coding RNA is binding with the protein, and then protein can getting close to the DNA. So this kind of complex will help form the binding between the DNA and the protein. And also it can change the stability of the protein. You know, a lot within the organism, there's ton of a protein. So protein can be easily degraded, right? But and then if you form the complex between the non-coding RNA and the protein, and this complex can help stabilize the protein. And then another feature of some non-coding RNA, it can function as the ribosome. So when I'm talking about the ribosome, which means RNA can work as a catalyst. For example, in the last chapter, when we're talking about the translational steps, we are talking about a very important catalyst to help to form the chemical bond between the amino acid next to each other. This enzyme, this catalyst called peptidyl transferase. Actually, this enzyme, it's a component of a large ribosome subunit, but this peptidyl transferase is it's a complex of the RNA and the proteins, and then RNA within this complex, they play a very important role as a catalyst. And then another type of the application of the non-coding RNA, it's a blocker. So and non-coding RNA can physically prevent a block cellular process from happening. For example, in the bacteria, antisense RNA, like it's type of a non-coding RNA, which this type of antisense RNA can complement to the mRNA. This kind of a complementary will help this non-coding RNA will bind to the mRNA. If this mRNA was bind, it's actually this kind of binding can work as a broker to prevent the mRNA go further for translation. And another type of the application about the non-coding RNA, it's called decoy. What's that mean? So which means some non-coding RNA can recognize other non-coding RNA and sequence it, and thereby preventing them from working. So like, for example, here, decoy non-coding, this is decoy non-coding RNA may bind to the this micro RNA. And then this type of a bonding can inhibit the translation of this mRNA. So so we can say this type of the decoy non-coding mRNA can prevent this microRNA to carrying out its function by these two binding together. So also this non-coding RNA can have an effect on chromatin structure and the transcription. Like for example, before we've been talking about non-coding RNA, it's in fact on the translational steps, right? And then those type of there are some type of non-coding RNA can also infect the first step, which is transcription steps. So here is an example. So this type of non-coding RNA is called a HOTAIR non-coding RNA, HOTAIR. This type of non-coding RNA. During the transcription steps, it can binding to two different type of complex. There's all like proteins, right? This green one and the brown one, they are all complex. Basically, they are like proteins, right? So this type of the non-coding RNA, they can bind to two different complex. And after like this huge complex formation, this complex will bind to the GA-rich region next to a target gene, right? It's gonna bind into this region next to this target gene, right? So after it's binding, and then this complex, these two complex, green and the brown one, will are very close to the histone here, right? Histone here, because it's getting very close because of the because of the non-coding RNA's binding and guidance function, these two complex are very close to the histone, so it can modify the histone, and then because the histone get modified, and then which will cause the inhibition of the transcription, and then they can also lead to the further change in the chromatin structure, and which also inhibit the transcription. Okay, so non-coding RNA can also have an effect on translation and mRNA degradation. Oh, this is too complicated for you. You're not going to talk about that. You can read about it. This is too deep. Let's talk about the mutation. This is very important. So mutation and DNA repair and the cancer. So first, we're gonna introduce the concept of mutation. What is mutation? So mutation, which means the change, there are changes in the DNA sequence of a gene. And then because of the change in the DNA sequence, so it will ultimately infect the gene function, like for example, gene expression. So, and then we're gonna introduce the type of the mutations which could happen in that would change the DNA sequence of the gene. So the first one is called point mutation. What's that mean? Point mutation, which means this type of mutation can infect only a single base pair within the DNA. Like for example, this is the original gene sequence, right? There's another one, but point mutation, which means it's gonna change one single base pair. So original, there was be T here in the DNA sequence. But here, we were, during the mutation, we're gonna change this T into the G. Here's another example. Before there's A here, but instead we change this A to C. So here, in these two examples, we only changed only one single pair. So we call it called point mutation. So point mutation could also involve the addition or deletion of single base pair to the DNA, right? This is more like base substitution, right? So different base will replace the old one, right? Another type would be addition or deletion. What's that mean? Like for example, before GGCG, right? Let's G after C. But here we insert A between C and G. That's called the addition of a single pair, a single base pair to the DNA sequence. And then another example would be G and C. That's G next to C. But we insert another T between G and C. That's also add another single base pairs to this DNA sequence. And because of this addition or base substitution, so the original DNA sequence gonna change completely, right? And so all these cases are called gene mutation. What's the consequence of the gene mutation? Gene mutations may infect amino acid sequence of the polypeptide. Let's see how it's changed. Like for example, so if this is the original DNA sequence, right? Each three single base will generate amino acid, right? So here, and then this DNA sequence will corresponding generate this amino acid sequence, right? For example, if we do point mutation, so change G here to C, that would be like that would be chance, right? Like for example, the amino sequence, which it's translated from this DNA signal won't be changed, right? Because 1, 2, 3, 1, 2, 3, 1, 2, 3, 1, 2, 3. Like for example, CCG, these three codon generate proline, right? But if we change G to C, maybe CCC can also generate protein. So let's say this type of a point mutation, we call it silent. Why it's called silent? Because even though the DNA sequence changed a little bit, like with one single base pair, but the translated to polypeptide chain didn't change at all because it's caused no change of the resulting the polypeptide chain. But look at another example. If we change here G into the C, but instead of alanine amino acid here, we generate the proline instead of alanine, right? Right? Before it's GCC, we generate alanine. Here it's CCC, we generate the proline instead of alanine. So we call this type of mutation, it's called the missense. What's that mean? Which means change one amino acid in a polypeptide chain. So then there's another type of change. What's that?